Welcome. Once again, welcome back. This is Dr. Hadi here, and you are watching Medical Glow by Dr. Hadi. Today's topic is DNA replication, and this topic is, of course, for the master level and advanced level. This is not a topic of 10 minutes or 5 minutes that usually some students are searching, but it contains a lot of detail. So if you want more detail about the DNA replication, you're welcome. I brought some question regarding the DNA replication that you will find the answer about the DNA replication. There are some special topics inside that DNA replication which most of students they want to get it. So maybe I will first, in the beginning of my lecture, I will recall these topics with you. Maybe you will understand. The first topic is, of course, DNA replication. Inside the DNA replication, there is a mechanism and type of DNA polymerase. DNA polymerase is the most important topic of this lecture. So we will study the types of the DNA polymerase as well as how it works. And then proofreading is another topic of today's lecture. Proofreading. Then the enzymes and some protein which are involved in that process, that will be discussed one by one. And what is nick sealing is a word that most of the students get confused from these two words. Nick sealing, what is nick and how it is sealed, how it is closed. And last but not the least, why DNA polymerase synthesize the new strand in 5 to 3 direction rather than 3 to 5 direction. So you will find the answer of this question in today's lecture. But what you have to do, you must watch the video from the beginning till the end, and then you will understand. But before I start, I would like to give you a suggestion that if you know the structure of DNA, you are welcome. You will get the lecture in a very good way. You will understand each and everything. But if you don't know the structure of DNA, the composition of DNA, you may feel some difficulties here. So I would suggest to go to that video which I have already recorded, that is the structure and composition of the DNA in English, because I already recorded in Urdu language as well. But you must type in English, and inshallah you will get this structure and composition, and then you will come back here. So come to the definition of the replication first. What is replication? Replication is a process in which one DNA molecule gives us two DNA molecules. In other words, we get two DNA molecules from one DNA molecule. The first DNA molecule which was present originally is called as the parental DNA. And these two DNA molecules which are formed from the DNA, these two are called as daughter DNA molecules. Once these two molecules are formed, there will be no parental DNA because it will be utilized. Today's whole lecture of 40, 50, 60 minutes will be consumed in this circle. We will study, we will discuss how the DNA is replicated. Okay, now this one more question. In books we use the word semi-conservative, So let me tell you what is meant by this word semi-conservative replication. Semi means half, mostly half. Once the DNA is replicated, if you see here how the DNA is replicated, first of all, the two strands are separated. It is not the detail, it is just the beginning. Okay, so first the DNA two strands are separated, the parental. This strand will act as a template. A template means single strand which will give us one more strand in the future. So this kind of strand is called as template, because with the help of this one strand we will get another strand. Therefore it is called as template. So in the future we will get a new strand in front of this strand. So this will become a complete DNA. And of course this strand will also act as a template, and in front of this we will get another new strand. So these two daughter DNA molecules, if you see here clearly, that one of the two strand is still the old parental strand, right? And this one is the new strand. This is not, this is the new strand. Because the old strand is still present, or we say conserved, therefore we use the word conserve, because half of the parental strand is still here and half of the parental strand is still here, therefore we call it semi-conservative, because half of the strand is still available with us, so we call it semi-conservative replication. Now next question is that why this process is occurring in the cell? What was the need for the replication of the DNA? What is the need? So many students ask this question as well. The answer for this is that you know that the body, in body, new cells arises from the pre-existing cell. So this is a cell, okay? This cell is going to divide into two cells, right? Cell number 1, cell number 2. And you know very well that this cell also contains DNA. Of course, in the form of chromosome. Chromosome, I will not use the word chromosome now. If chromosome is present in the cell, definitely DNA is there, because DNA is the part of chromosome. So let's suppose this is the DNA present in the cell, and when the cell is divided into two parts, what would you do if you perform justice, if you provide justice? So you must transfer equal amount of DNA to both the cells. So if you have one DNA strand, this one DNA strand will be transferred to this cell, and there will be no DNA left to enter into that cell. Therefore, what we do, we will duplicate the DNA, so we will get two DNA. So one DNA replicated into two, so two DNA we got. Now what will happen, one of the DNA molecule will enter into that, and other DNA molecule will enter into that cell. And now look at that, here you can see this is one DNA, okay, and this is another DNA. So two DNA molecules are now formed, and one of the DNA has been given to one cell and the other DNA has been given to another cell. We just provide them a full justice here, right? So this was the answer of the question why there is a need of DNA replication. Okay, and one more thing you must remember, that the DNA contain complete information of the organism, right? Each and every information is contained in the DNA molecule. So if these two cells must be given the required information so that they could perform in the future. Every cell is totally dependent on the DNA. Every cell is totally dependent on the DNA. In other words, the information of the whole body information are present in this DNA molecule. So that is why this is a very, very, very special molecule. It must be transferred to each and every cell. Okay, dear students, now we will come to another topic, that what are the enzymes or proteins which are used in that process. So there are some enzyme first, okay, then we will tell you something about protein. The enzymes, I can write them with the word HLPG, right? LPG. So H is for helicase enzyme, this is for helicase enzyme. L is for ligase enzyme, and P is for primase enzyme, and G is for gyrase, or you can call it topoisomerase. Here the helicase is also called as the DNA B protein, right? So these are some enzymes. There are some enzymes which I can teach you here just right now, but other enzyme we can't study it now We can't study them right now, but at the end of the lecture we will study them, like topoisomerase, this is also called as gyrase, primase enzyme, and ligase enzyme, and helicase enzyme. This is the enzyme that we can easily understand it. What is the helicase enzyme? And the king of all those enzymes is the DNA polymerase. There is one enzyme called as the DNA polymerase. DNA polymerase. Okay, this is the DNA polymerase. DNA polymerase, there are types, 3 types. Mostly in books of Leninger, they said there are 5 types, but mostly there are 3 types of the DNA polymerase: polymerase 1, polymerase 2, and polymerase 3. We will study these 3 enzymes in detail. But we start with the ligase enzyme first. With the helicase enzyme first. What is helicase enzyme? This is the enzyme that unzip the DNA molecule. We know very well that a DNA consists of 2 strands, okay? And if I write here 5, 3 and 3 to 5, these things are already mentioned in previous lecture, 5, 3, 3, 5. And the 2 strands are linked together by the help of the hydrogen bonds. Okay, so if you want to duplicate the DNA, first of all you must separate these 2 strands from each other. So who will come? That this is the helicase enzyme. The helicase enzyme, what it will do, it will unzip the DNA so that it will break down these hydrogen bond and the 2 strands will be separated. Now, come to the protein. What are the protein here? This is one DNA A protein, DNA B protein, DNA C protein. One more thing is here, that is a single strand binding protein. We call it SSBP, single strand binding protein. Okay, single strand binding protein, when the DNA strand is separated, let's suppose this is the DNA strand, right? 2 strands are separated. Okay, let me write it in one color. That is, let's suppose this is, okay, that is the DNA, right? Let's suppose this is the separate, these 2 are the separated strands. There is a chance that the 2 strands will recombine, join together back, and it will disturb the whole process. We will not let these 2 strands to combine together, because we are going to use each strand as a template, and we are going to make a new strand in front of each strand. Therefore, these 2 must be separated. And who will prevent these 2 strands from joining together? That is the single strand binding protein. So SSB protein will bind here and it will prevent the rejoining. Dear students, here is the DNA B. DNA B is a protein. It is also called as the helicase enzyme. Its function is already discussed. It will unzip the DNA strand. Now come to the DNA A. DNA A is another protein that we will discuss it in the initiation phase of the replication. It will denature the DNA. A slight denaturation of the DNA, it will just denature it, and a small replication bubble is made by this, and then the whole process will be given to the helicase, and then the helicase will start unzipping. So the initial unzipping process is done by the DNA A protein. The initial process, and then helicase will start unzipping the DNA. So you will also get these things in the lecture. So now let's start the replication process in detail with the stages. The replication process has 3 stages for us. It says 3 stages. This is the most important diagram that we are going to discuss. 3 stages. Stage number 1 is the initiation. Okay. Stage number 2 is the elongation. And stage number 3 is the termination. So we will discuss the whole topic in these 3 stages. Initiation means start. So first of all, this is the DNA. Okay, this is the DNA, and first of all, the replication process start or begin with some specific point in the DNA, right? That is called as replication origin, right? We call it origin of replication. So if you want the process to be started, first of all you must find the origin, the place where the replication is started. It is not possible that you start replication from anywhere in the DNA. There are specific points, specific places on the DNA called as the origin of replication. The origin of replication contains some sequences. They contain specific sequences. Sequences of what? Just like AT. Usually you will find a repeated sequence of adenine, thymine, AT. So that is one identification of the point. If you want to search that, where is that place, what is the identity of such kind of place. So if many AT, repeatedly AT sequence come, this is identification for the origin of replication. Remember one thing more, I did not tell you in the beginning that the replication can be studied in the prokaryote as well as in the eukaryote. So now we are in the prokaryote, right? We are studying this replication process in the prokaryote, right? So this is a specific sequence. We call it origin of replication. Let's suppose this is the site. That we say this is the origin of replication, OR, origin of replication. Now what we have to do, we have to separate these 2 strands, right? So after the identification of this site, a protein will be attached to this site called as the DNA. The DNA, as I already told you, that we will discuss each and every enzyme protein in the lecture. So now here comes the function of DNA. D, small n, small a, DNA. Don't worry, this is not the DNA. This is just a protein who is working on the DNA. So this is a protein called DNA. This protein will attach here. What this protein will do, it will denature the DNA. You know, denature means it will break down the hydrogen bond. Let me write it a little bit in more larger size. Okay. Let's suppose this is the DNA. Okay. This is if I write just straight, it will be more good. If I write just straight lines, okay, 1 line and 2 lines, right? These are the nitrogenous bases that are attached with each other. Okay, this is DNA, 5 prime, 3 prime, 3 prime and 5 prime. Suppose this is the origin of replication. This is the origin of replication, a specific area, and here we have the DNA A protein. This is the DNA A protein, right? And what will happen at this area? A small small bubble-like structure will be formed like this. A small bubble-like structure will be formed like this. Okay, this is a small bubble-like structure will be formed like this. Still we have 5 prime, 3 prime, 3 prime and 5 prime. This bubble is actually it was straight like this, but now when the hydrogen bonds are broken, so what will happen, the nitrogenous bases which were attached with each other, they will be exposed. Now here guanine, cytosine, thymine, guanine, these are exposed, and they also will expose cytosine, adenine, guanine and cytosine, right? There is no hydrogen bond between these two. Now this is called as replication bubble, right? This is called as a replication bubble. Remember, this was only made possible by the help of the special sequence called Origin of Replication, and who identified, that is the DNA, a protein identified it, attached here, and it has just removed the hydrogen bonds, and a slight replication bubble was formed. According to the book of Satya Narayan, that in the eukaryote you may find more than one replication bubbles like this, means like this. This is okay, this is this is the DNA. You may find one replication bubble, 2, right? You may find many replication bubbles. So the question is, why there is a need of many replication bubbles? The answer is that in order to make the process speedy, so that is why many replication bubbles are there. We are not concerned with those many replication bubbles. We are concerned with only one replication bubble like this. What will happen? As I already told you, that one strand acts as a template and the other strand will also act as a template. One strand will act as a template to make another strand. So how another strand will be synthesized? Here like this. This will be the new strand in 5 to 3 direction. In here another strand will be synthesized in the opposite direction, right? In the opposite direction, 5 and 3. So remember, this is a bidirectional replication, bidirectional synthesis. One strand is synthesized in that direction, the new strand. Another strand will be synthesized in this direction. Okay, so then what will happen? You know very well that this process will be made continue, because the helicase enzyme will will open these two strands, and the process will go on, right? The process will go on. The two strands will be separated and the process will be go on and continue. In this way we will get the two strands. Now the most important part of this lecture that students feel so much confusing, that how the new nucleotides are attached, and how these are given a new strand. Okay, slight disturbance was there. Okay, so the main problem for the student is that how the new strand is synthesized. This is most important. So come to the second part. This was just initiation. Now the third part is called as the elongation. Okay, now the second part is called as the elongation. In elongation process, let's suppose this is one replication bubble. If I just erase this part, it will become like this. Okay, now this is called as the replication fork. Mostly in lectures we write it like this. Okay, we write it like this. This is now called as the replication fork. Okay, this is called as the replication fork. We will study only one replication fork, and it will lead us to the understanding of the whole DNA. Okay, so in elongation we have this replication fork, right? This was a replication bubble, now we made it a replication fork, right? This is one replication fork. Of course there is another replication fork as well, so we will not discuss this. So we will discuss only one replication fork. If it is 5 prime, look at that, if it is 5 prime, so then this is 3 prime, you know very well. If it is 3 prime, then this is the 5 prime. Okay, now an enzyme will come which is called as the DNA polymerase, the DNA polymerase enzyme. This is most important enzyme that will add a new nucleotide to this strand, and it will make another new strand. But there are some problems with the DNA polymerase. The problem is that the DNA polymerase need something to start. It cannot start the process by itself. And one thing more, we will discuss the types of DNA polymerase later on, but now I'm not going to tell you the types of DNA polymerase. So we have one DNA polymerase. It need a primer. You know what is primer? Is a kind of something that give a start to the process. It's a kind of something that give that help the DNA polymerase to start the process. So what it is really? A primer is part of a piece of the RNA, a small segment of the RNA, right? That contains a free 3-OH. Let's suppose this is a small piece of the RNA, contain adenine, guanine, uracil, okay, and guanine and cytosine. Let's suppose this is just a small piece of the RNA, right? This is a small piece of the RNA. It's not DNA, because you can see there is uracil, and uracil indicates this is RNA. So this is a small piece of RNA. That small piece of RNA which contain a free 3 end. Of course it contains 5 end. At 5 end you will see a phosphate is there, and at the 3 end you will see OH. Don't worry about why this is 3. You know very well whether it's a DNA or RNA, there will be 3 end and 5 end. So at the 5th end there will be phosphate, and at the 3 end there will be a free OH. This is the most important thing to tell you, that at the third carbon there is a free OH. So this small piece, according to some books, 50 to 150 nucleotides is called as primer. And who make this primer? There is an enzyme called as the primase. As I already told you that we will discuss enzyme during the lecture. So there is an enzyme called as the primase enzyme, right? Primase enzyme. The primase enzyme make a primer, and that primer is attached with the DNA strand, and then the enzyme DNA polymerase start its function. What is now the function of the DNA polymerase? The function of the DNA polymerase is to polymerize. To polymerize mean to add the nucleotide, the fresh nucleotides. We have many nucleotides, just like I have just mentioned, only one here, right? There is one nucleotide. Look at this is one nucleotide, right? This is sugar, adenine nitrogenous base, and there are 3 phosphate. The nucleotides in the beginning have 3 phosphates. So we call it D. D means DNA adenosine triphosphate. Similarly, we have D guanosine triphosphate. We also have D cytosine triphosphate. We also have D thymine triphosphate. So these are the nucleotides. Most of students, they think that the nucleotide contain only 1 phosphate. No, it is wrong. During the synthesis of the DNA, each nucleotide contains 3 phosphates. When 1 nucleotide is added, the 2 phosphates are released in the form of pyrophosphate, right? So we use the word pyrophosphate, is actually 2 phosphates that are released. So what is the function of a DNA polymerase? And dear students, I spent almost a lot of time and that's this DNA polymerase. There are a lot of things, there are a lot of information attached with the DNA polymerase. But the most important point that I got for you is that the DNA polymerase is a complex. Complex is in a sense that it has types. 5 types are there. 1, 2, 3, 4, 5. 1, 2, and 3 are most important. Among these, the DNA polymerase 3 is most important because it is the DNA polymerase 3 that make a new chain. One thing more, the DNA polymerase 3 enzyme occupy the whole place, both the 2 strands, and at the same time the 2 strands are being synthesized by 1 DNA polymerase 3, not from the different DNA polymerase. So let's start, because we are in the second process, elongation. As I told you that the DNA polymerase enzyme can only start when the primer is there. So first of all, we will add a primer. Let's suppose this is the primer here, a small piece of RNA that we have. Here is the primer. Let's suppose this is the primer. Okay. G and C, and in place of T, A, there is thymine. So thymine is not in RNA, so we'll write it as uracil. This is just a primer now, right? Primer. And remember what I told you, that a primer contain an OH group at the 3 end. So where you will expect the 3 end? Here? No. The 3 end is always here. That is the 3 end. And this will be the 5 end. Because remember, if at the original strand the 3 is at the left side, so the OH of the primer will be on the right side. Remember, this is just, most of students get confused. So you can memorize by this, that if this is the chain, the 3 end is there, the 3 end is at the left, so the OH of the primer will be on the right. And if let's suppose this is 5. So if you want to add a primer here, the OH of the primer will be on that side or on that side. Of course, opposite to 3 end. So if it is the 3 end is here, so the OH will be at that left side, right? Here the OH is on the right side, so here the OH will be on the left side. This OH will serve as a site for the new nucleotide. When the fresh nucleotide comes, it will make a bond with this OH. This oxygen is a very good nucleophilic center, nucleophile. Okay, what is mean by this nucleophile or nucleophilic center? You know very well, nucleophile is something that love positive, means something that contain a negative charge, something that contain an electron, something that contain more electron, something that has the tendency for the positive charge. So anything that has the tendency to get the positive charge is called a nucleophile. So this OH is a very good nucleophilic center. So we will expect a nucleophilic attack here. Okay. The DNA polymerase will take one nucleotide. Let's suppose this is nucleotide. and it will attach the nucleotide here, because the nucleotide contain phosphate. So what will happen, these two phosphates PO4, these two will be removed, and this whole nucleotide, which is adenine, but here is thymine, so in front of thymine we must have adenine, so adenine nucleotide will be attached here. So I'm going to attach what, a new nucleotide as a adenine. I will not show these sugar phosphate here because there is no need to show these, but now look at that, the adenine has been attached, and of course this OH will serve as a bond, as a phosphate bond. So there will be a phosphate bond between that uracil and the adenine. So again one phosphate served as a bond is formed now here. Now then what will happen, then it is guanine. So one thing more, if the adenine has arrived, adenine has also have a 3 OH group. So here we again have OH group. So another nucleotide, can you tell me which nucleotide will come in front of guanine? Of course cytosine will come, because we know very well always in front of guanine cytosine will come. And dear students, I got a very important special answer for this. Many students, they ask, they sometimes tell, sir, why guanine is always paired with the cytosine and adenine is always paired with thymine? You know that everything is done here by the DNA polymerase. So the answer behind this is the geometry. In the active site of the DNA polymerase is only suitable to be fit for this kind of geometry. Means this is a special geometry of guanine cytosine, a special geometry of adenine thymine. There is the shape these two attain can be easily fit in the active site of the DNA polymerase. But if adenine comes here, guanine is attached with adenine. Although we saw that hydrogen bonds can be formed between guanine and adenine, yes, it is possible. But once the guanine and adenine are bound together by the hydrogen bond, they will not get the same shape. Their shape is something like this. So when the shape is changed, there is no place, there is no such a site on the enzyme to occupy this one. Therefore, it is not possible that guanine is attached with the adenine. It will come under the heading of some error. There are some error, okay? There are some errors sometime. It happens that wrong bases are attached. There is something else, we will discuss it. But this is the answer, that why guanine is always with cytosine and adenine is always with thymine. This is because the credit goes to the active site of the DNA polymerase enzyme. Okay. So the next nucleotide will again attach here. In front of guanine, we will get a cytosine. Of course, this OH and the phosphate, they will make a bond, which we will call it phosphodiester bond. So slowly we will make a new chain. We will make a new chain. This is the chain that has been formed. Now can you tell me what is going on here? This chain is moving in that direction, right? This is going towards the replication fork. You know very well this is the replication fork. These are the single strand binding protein that will prevent these two strands to join together, and this is the DNA helicase. This helicase is moving forward. It is just separating the two strands. So the strand which is moving towards the replication fork is definitely called a continuous strand. This is called a continuous strand, right? This is called a continuous strand. In other words, it is also called as the leading strand, because it is synthesized continuously, it is moving forward. It is called as a leading strand or continuous strand. And this is so simple, this is so easy. Now, if you just tell me what is the direction of this new strand, what is the direction of this new strand? So remember, the credit goes to the DNA polymerase, that the DNA polymerase always make the new chain in 5 to 3 direction, in 5 to 3 direction, in 5 to 3 direction. Look, this is the 3 end. The primer is always attached to the 3 end, so the DNA polymerase will start from the 3 end, and the new strand will of course be 5 end. So this will be the 5 end. This is so simple. If this is the 3 end, the OH was a 3 end, definitely the phosphate of the 5 end attached here. So this is the 5 end and 5 end, 5. Of course, this is again the 3 end. So we already discussed this, that every nucleotide that comes has a free 3 end which contain OH group. So this is the 3 end. Of course, the beginning will be the 5 end. So the new strand has been synthesized in 5 to 3 direction. This is integral part. This is much important, that the DNA polymerase can only make the new chain in 5 to 3. 5 to 3 is only possible because the situation is like this. But the DNA polymerase never make a chain in 3 to 5 direction. However, the breaking can be done in 3 to 5 direction. The breaking can be done in 5 to 3. But the making process is always in 5 to 3 direction. Okay. Now the special thing is here. Come this strand. Look at that. This is the 5 end and this is the 3 end. We want to make this strand as well. Again, we must make the new chain in opposite direction. Means if this strand is 3 to 5, so the new strand must be in that direction, right? In that direction. 3 and 5. Okay. But there is some special thing, okay? In this case, listen, in this case, if I attach the primer here, let's suppose this is the primer, okay? This is the primer, okay? Primer. This is the primer. Where do you expect the OH will be? The OH, would you place the OH here? Or you will place the OH here. Where? I already told you that as the OH was at the opposite side of 3, this is 5, so 3 will be here. If this is 5, this is 3. If this is 5, this is 3. This is 5, this is 3. 5, 3. If this is 5, this is 3 end. If this is 5, this is 3 end. So if this is the 3 end, the OH must be at that side of the primer. So in this case, if the OH is of the primer is on that side, definitely the DNA polymerase enzyme has to make a new nucleotide here. We don't have such thing here. So we must make the chain in that direction. The exposed part is available here. We must make this part, not this one. So the primer will be placed not here, the primer will be placed here, something closer to the replication fork. Yes, we will place it closer to the replication fork here. This is now the primer. Okay, this is now the primer, right? And yes, if this is a 3 at the right side now, the primer's OH will be at that side. Okay, always opposite. Now what will happen? A DNA polymerase will take the nucleotides and will attach here. So I will not discuss it again now. The polymerization process is there. So in front of guanine, we will have cytosine, and in front of cytosine, we will get guanine. Between the cytosine and guanine, there will be a phosphodiester bond that we will not discuss now. There will be a phosphodiester bond. Between the primer's OH and this cytosine, there will be a phosphate bond. So a bond will be there, and here Another nucleotide will be placed here, another nucleotide will be placed here, another nucleotide will be placed here, right? The process will be continue. Okay, continue. Another process will be here and here. Okay. So now you tell me the new strand's direction, where it is going. This time the new strand's direction is in this direction, it means away from the replication fork. So the new chain which is synthesizing in that direction is called as the discontinuous strand. Discontinuous strand. It is also called as the lagging strand. It is also called as the lagging strand. Okay. Is this enough? No, this is not enough. Now what will happen, that still we have this portion is available to us. Okay, still we have, and this is we have more nucleotides here. So more nucleotides here. So what will happen? Another primase enzyme will come. It will make another primer towards the replication fork. So another, because the two strands are separating. So we will make another primer, another P. Primer is attached here by the help of primase enzyme, and we will make another chain. This will be another chain, right? Okay. So another chain will be synthesized again. In which direction? In 5 to 3. Dear students, look, if this is the 3 end of the primer, definitely this is the 5 end of the new chain. If this is the 5 end, this must be the 3 end. So look, DNA polymerase, whether it is here or here, it always synthesizes the new strand in 5 to 3 direction. So again it is in 5 to 3 direction. So this is 5 to 3 direction. This is again 5 to 3 direction. What you can see here, this chain is being synthesized in pieces, not continuous, just like this one was synthesized continuous, but this one is synthesized in pieces. This is one piece, this is another piece. These pieces are called as, of course, one piece is separated from another piece by the help of primer. So later on we will remove this primer, but these pieces are called as Okazaki fragments. These pieces are called as Okazaki fragments. Okay, and the chain length is 1000 to 2000 in prokaryotes. In case of prokaryotes, the length of the Okazaki fragment is 1000 to 2000, and in case of eukaryotes, 150 to 200. In case of eukaryotes, 150 to 200. This is just the length of the Okazaki fragments. Okay, so one thing more, I would just make this diagram a little bit beautiful for you people, because there are so many things here. Okay, I'm going to erase this one. Okay, suppose that the whole DNA molecule is now being separated, and the process is called as elongation, because you know very well that the chain length is being increased, so this is called as elongation. Let's suppose the process is being ended. Now the third process, or the last process, is called as the termination. Okay, the third and last process is called as termination. How that process is terminated? They say, in the DNA, just in the initiation phase, there is specific sequence was there called the origin of replication. For termination in the DNA, there is specific sequence. There is specific sequence here. Again, once again, there is specific sequence. They are called as ter. Of course, terminus. That specific sequence is called as the terminus, right? There is another protein called as the TUS protein. So TUR and TUS. This protein specifically binds to that portion of the DNA called as the terminus region. Once the protein is attached at the terminus region, the process will be automatically stopped, because it will, what will happen, it will stop the replication fork. You know that the replication fork is moving forward. You are opening the zip, it is moving forward. So when the terminus region come, and it is a specific sequence, a protein will be attached. Once the protein is attached here, the replication fork can't access through that point, so it will stop here. So the whole process will be stopped here. So the termination was so simple. That was so simple. Nothing was there. And now let's suppose this was one DNA strand completely separated. This was another DNA completely separated, and in front of one DNA we got one continuous chain, right, in 5 to 3 direction, and here we got another chain, but this was in pieces. This was 1 5 to 3 direction, okay, and here was another chain again in 5 to 3 direction, and there was another piece in 5 to 3 direction, okay. This was 5 to 3 direction. What was there in between these two strands? Definitely you will say, sir, there was a primer, okay. And so here is the primer P, here is again the primer P. Dear students, now it is time to discuss with you people the type of enzyme we will discuss in the DNA polymerase. The function of the DNA polymerase 3 was done. That was the polymerization process. One more process of the DNA polymerase 3 is the proofreading. What is mean by proofreading? A proofreading is that, let's suppose sometime, according to the book of Leninger, that sometime a nucleotide undergoes a small tautomerism, a kind of isomerization. And so what happened, a wrong base comes, let's suppose in place of C there comes thymine. Okay, this is wrong base. Sometime it happens, right? So the DNA polymerase has the capability to check this error and to make it okay again. What will happen? The DNA polymerase 3 has its cleavage action as well. Means it can break nuclease. We call it the exonuclease action. What does it mean by exonuclease? Means the breakdown of the DNA is called as the nuclease activity. So the DNA polymerase at one side it can make a new chain, and on the other side it also has the ability to break down a chain. So it can break the chain in 3 to 5 direction. Means if you are making a chain in 5 to 3 direction, okay, if you are making a chain in 5 to 3 direction, the DNA polymerase has the ability to break it down in 3 to 5, like in this direction. So the DNA polymerase is moving in the forward direction. When mistake occur, let's suppose a mistake come here. So what will happen? It will stop. And just lyse it, break down, and during breaking the wrong nucleotide, let's suppose what we discussed in place of C here, C we got thymine here. So what will happen? The DNA polymerase 3 will remove this nucleotide, and in place of this it will put a guanine. Once it is okay, it will move forward. So this is called as proofreading. Means that let's suppose DNA polymerase is moving in that direction, mistake occur, and what it will do? It will reversely cut down, like it will break down the DNA. So in that direction, it just break down, remove the nucleotide, make it correct, and then it will go in the same direction. So this is called as the proofreading. Polymerization and proofreading both are done by the DNA polymerase 3. Now we will come to another topic, that is the DNA polymerase 1 and the DNA polymerase 2. Here is the DNA polymerase 1 and the DNA polymerase 2. The DNA polymerase 2 is involved in the repair. Sometimes if some damage occurs to the DNA, it will just repair it. So there is no other explanation for it available. But the DNA polymerase 1, this is another enzyme, DNA polymerase 1. It has two functions, two most important functions. The one is the proofreading and the replacement. Okay. The replacement is first of all. Okay. What is mean by replacement? If I just raise it like this, suppose we have, this is the strand, right? And we have a primer here, right? So let's suppose this is guanine and cytosine and guanine and cytosine, right? There's a new chain. We have primer here, attached primer. So primer is here. C, G, and again, let's suppose the red color is primer. So the DNA polymerase will come. It will remove this primer. So once this primer is removed, so you know very well in the DNA, for the G there must be cytosine, and for the G there must be cytosine, and for C there will be a guanine. So once the primer is removed, so the DNA polymerase 2 will place what? It will add another nucleotide, cytosine, and then that's guanine, and here again a guanine. So this was a kind of gap left. When the gap which is left by due to the removal of the primer, this gap must be filled. So who will do this? The RNA, the polymerase 1. This is called the polymerase 1. The polymerase 1 activity, DNA polymerase 1 activity. So wherever you find a primer, the primer will be removed, and in place of primer, the nucleotide sequence will be placed, especially in the discontinuous strand, in the lagging strand. So one most important thing is that a small gap, very small gap, will be left, called as the nick, as I told in the beginning. Nick, N-I-C-K, nick. What is nick? This is a continuous chain, and this was just a chain that was made now in place of primer. The primer, the nucleotides in place of the primer and the new chain, there is no bond. There is no bond. That place where no bond is present is called as a nick. So this is a nick. You know very well that if it is one nucleotide and this is another nucleotide, if this is the 3' end, this is the 5' end, so between the 3' end of one nucleotide and the 5' end of another, there must be a phosphate and there must be a bond. Okay, this is called as phosphodiester bond. This phosphodiester bond is called a nick. It is left. Okay, so we must make the bond here. So the process is called as nick sealing. So who will come for it? Another enzyme will come for it, called as the DNA gyrase. I promised you that we will study this DNA ligase, not so DNA ligase. DNA ligase will seal the nick. A DNA ligase will make a bond between the 3-OH and the 5-phosphate. DNA ligase will just help to make this bond, and then it will remove, and it has done its duty. So there is another also a mechanism for the DNA ligase. First, the DNA ligase contain amino group, it attaches 2 phosphates with it, and then come here. So there is no need to discuss this. Simply, the phosphodiester bond between the polymerized chain and the freshly made chain in place of primer is done by the help of the DNA ligase enzyme. So this is a nick sealing enzyme. Now what left? We got each and everything. Proofreading was there. And so one thing more, that the DNA polymerase 1 has 2 functions, as I have already mentioned there. Number 1, it has the ability to synthesize a new chain in place of primer. This is called as the polymerization function of the DNA polymerase 1. Number 2, it has the ability to proofreading. Proofreading, as I already discussed with you people, proofreading is done by the polymerase 3, and more proofreading is done by the polymerase 1. So polymerase 1 has 2 functions. Number 1, in place of primer, add the nucleotides. Number 2, proofreading is there, and the ligase was done. Each and everything is done. I think. Let me tell you, is there anything left? Okay. I hope each and everything you just got this thing. No question left here with me. Maybe if any question left, please write in the comment box. I will be ready for your question. I hope you enjoy that lecture. It was lengthy, but what I have to do, I have to deliver my best. So thank you for watching the video, and have a good day. Thank you, and bye-bye.